are different types of even nanoparticles. The different institutions they are working on the nanoparticles, how we can manage these pests. Then lymphatic paresis. It is one of the important vector-borne disease. Paresis is caused by the parasitic worm, like intestinal worm, Ascaris lambricoides or Dracunculus medinensis. It is Wucheria, then Croftiae, then Bugia. This type of species they are reported from even Himachal Pradesh. And the world map of the lymphatic paresis, you can say here is the non-endemic, then not started, ongoing, means there is surveillance from the world, and we have the different types of programs for the surveillance. This is the life cycle of Wucheria bancrofti, means how it infects the human being, how it means it is transmitted by the mosquito, it is transmitted by the armadillos, from one person to another, from the unhealthy, from the infected to the healthy person. Then these are the different hosts of these encephalitis. These are the hosts. Some of the hosts they are the amplified host, means they amplify, they multiply this pathogen, that is the Wucheria bancrofti. Then undead end host means they cannot multiply further. Then vector. Then spread of this Japanese encephalitis, this uninfected mosquito bites the pigs and waterworms, means they have the different types of hosts, so therefore the host play a very important role. This is the trend of Japanese encephalitis in India, state wise burden. इसमें आप देख सकते हैं कि there is a highest cases in the Assam. Then Japanese encephalitis in Himachal Pradesh. This is rare in Himachal Pradesh, but it is recorded. And there is a total of 8 cases were reported, with 7 confirmed through ELISA. From the different tests they are confirmed, and the current status of 2024 to 25. This disease has not reported as a major disease from the Himachal. This is the fortunate part of this vector-borne disease. We are working on the different aspects of these vectors, like morphological integration and molecular characterization of the Eastern tiger mosquitoes. We have prepared the phylogeny of different mosquitoes of this vector. Then I'm going to conclude with 2 minutes. So please. Survey of mosquitoes in Himachal. We have surveyed the 6 districts of Himachal Pradesh, means intensively we have surveyed. Then we have surveyed the larval adult population of this vector. Then mosquito species identified like Aedes aegypti, Aedes albopictus, and Armillaria sublacetus. Means these 3 important species they are recorded from the Himachal Pradesh by our research group. Then identification method we have identified on the basis of morphology through SAM-FAM, and we have identified the different species. We have identified the different types of this is the scenario of the population from the 6 districts. Then we have taken the samples. These are the different types of species recorded by our group. These are the larval stages, these are the different identification features, these are the alpha structures. Then the mosquito richness is found in these districts. These are the morphological differences, we on the basis of morphology we identify. Then we have the SAM investigations, we have identified on the basis of this electron microscope. We have recorded the different types of sensilla from the mosquitoes. They have the different types of morphological changes, and these changes they are responsible for the due to the geographical conditions or due to the climate change. There are different types of non-olfactory sensilla and olfactory sensilla. These are the different types of sensilla, and the function. This is very important. It means what is the function of the sensilla and how we can interfere during the management practices. How we can control the different types of sensilla. Then these are the types of sensilla, these are the types of under the scanning microscope. So we have to means focus on the surveillance, and we have to identify the features which are responsible for the attraction or for the host finding. Thank you very much.
Thank you very much, sir.
My thanks to Mr. lab groups, research scholars for completing this work, and I also thanks my university, Central University for funding, and DSP and DHR for funding the projects. Thank you.
Thank you very much, sir, for your valuable words. Before today, whenever I saw a mosquito, I always go for killing. But now I realize that how many species are available in the in the animal world of the mosquito. It was very wonderful lecture, sir. Thank you very much. Now it is the time to facilitate two very renowned personalities which are having it with us. I would like to invite our worthy principal, sir. Please come on rise and facilitate Dr. Arup Giri, Baba Masnad University brother, Dr. Arup Giri. Give a big round of applause for his attendance. We are very glad to honor such a personality we are having today. मुझे पता है कि अभी वीकनेस हो रही है, सुबह से बैठे हुए हैं, लंच नहीं मिला है, पर फिर भी तालियों में कोई कमी ना की जाए प्लीज. Because the clapping is the actual honor of any guest or any speaker. By moving forward, the next we are Yunus Ahmed Hassan, son Baba Bhag Singh University, Jalandhar. Welcome you, sir, in our premises, and we are here to honor you for your valuable presence. So moving forward, now I would like to call Professor Rakesh Kumar, CUHP Dhanshala, for a word of thanks. Please, sir.
Good afternoon, coming all present here.
हाँ इसके बाद मेरी है प्रेजेंटेशन.
On the behalf of organizing
मैं आपको
committee,
दूंगा.
I would like to thank
अभी दिया नहीं है. थोड़ा सा एडिट करना.
speaker.
क्या करना है? थोड़ा सा एडिट. हाँ.
Professor
एडिट करने के लिए
Can he mention about the bio
दिया नहीं.
and the
मैं ही
biopolymer and their
करूँगा.
application
क्या
of
मैं बता
biological sciences?
दे दूँ.
How this biopolymer is helpful in agriculture and in the medical sciences and drug delivery. So, sir has delivered a very wonderful lecture. Thank you very much, sir. Second, our eminent speaker, Professor Sunil ji. Sunil ji mentioned about the mosquito diversity and their ultrastructure. Mosquito they adapt from different altitude and their diversity has changed from altitude to altitude, and they are the sensina. Like our nose, they have power of smell. Like their sensina has the power of smell. So they have adapted in the different condition in different ways, and in different, they have changes their adaptation in climatic conditions. So I would like to thank to our eminent speaker, Professor Sunil Thakur. I would also thank to who chaired this session, Professor Patania ji, Professor Mehta ji, Professor Rajesh ji, who chaired and co-chaired this session. I would also thank to the participants who listened for their presence. I would thank you very much.
Thank you.
ये मेरा है.
Thank you very much, sir, for your valuable words, and thank you. Now it is the time to reboot ourselves. Reboot. Why I am saying reboot? Because I am the I am from the department of the computer science, and rebooting means re-energizing ourselves. So please move forward for the lunch, and I hope that sharp at 3:30 we will get together for the next session. Thank you very much. One more time, the oral presenter who will present their presentation kindly contact with the technical and diplomacy for their PPTs. Thank you.
हो गया सी वो कर दे बंद नहीं कट